Now that we've got, okay, now we're starting recording. We got results from 4 of the 6 or 7 urchins that we injected. And these are what the eggs look like. They're slightly yellow. It's not easy to see with this camera, but I can see that yellow is made up of a bunch of small dots. These are maybe less than half a millimeter, but still large enough that you can see the individual dots with the naked eye. In this species, they happen to be yellow. In the ones that we use in the spring, Strongylus introtus, they tend to be more of a pinkish red. I'm not sure why that's significant, but there you go. And there is what the sperm looks like. It's that milky stuff. And there's some more. So we got 2 males with good sperm samples. And I might be tempted to just dump the sperm right in with the egg. The problem is that there are ridiculously large numbers of sperm in here. And basically, if I do that, each egg would be attacked by 1 million sperm maybe, and that would overwhelm the egg's defenses. Now, what do I mean by defenses? I mean that it's bad if an egg gets fertilized by multiple sperm. If you don't have 1 egg and 1 sperm, you don't get a diploid nucleus, and cell division can't happen properly. So to avoid that, the egg has 2 ways of blocking polyspermy, which is what you call it with multiple sperm hitting. The first is called the fast block, and that is a wave of electrical polarization that sweeps around the cell the minute the sperm has penetrated the membrane. That is practically not quite instantaneous, but it's very fast, but it only lasts about a minute. The second block is the slow block to polyspermy. And what happens is a bunch of tiny vesicles right under the egg plasma membrane release substances into the extracellular space that absorb water, harden, and cause a membrane that surrounds the egg, the fertilization membrane, to lift off. And that takes a few minutes, but that's a second physical block to double fertilization. And so the first one happens very fast, but it doesn't last long. The second one takes longer, but is permanent, and it creates a firm shell around the developing embryo. But if I were to dump concentrated sperm in here, there would be so many sperm that they would just completely overwhelm that poor egg. And every egg would get fertilized by many sperm, and you'd end up with very abnormal embryos. I've seen it happen. It really looks strange. Cells don't divide evenly, so you have embryos made up of cells of many sizes, and then, of course, they never develop further. They just die. Remember that in the wild, this would all be happening in the ocean, and however many sperm a male releases, they're going to be diluted pretty quickly simply by the action of the currents. What we have to do is get a clean pipette. And I'm going to take about half a mil, let me see, of this sperm. That's about half a mil. And dilute it in this beaker. That's 50 ml of artificial seawater. And now I can add that to some eggs without the eggs getting hit by so many sperm that they all end up with multiple fertilizations and very abnormal development. So I'm going to take the eggs and add some artificial seawater to dilute them a little bit. And I'm going to make a wet mount. And I'm going to stop recording right here, and we'll come back when I've got it under the scope.